Yep, thank you. Good morning. It's a pleasure to be back here in Birmingham. I'm Liam Crowley. I'm currently a postdoctoral researcher at the University of Oxford, working at Wytham Woods. But before that, I did my PhD here at BIFoR, looking at the effect of elevated CO2 on insect diversity at the Millhaft facility. So yeah, apologies. I've been really ambitious with the amount that I'm trying to cover today, so I'll have to rattle through a little bit, but hopefully we'll get there. So I shouldn't have to convince you that forests and woodlands are important. We know they're globally important carbon sinks. We also know they're important for water regulation, but also really important for housing biodiversity. What I will hopefully convince you is that actually, when we're thinking about biodiversity in terrestrial ecosystems, we need to be thinking about insects and other invertebrates. They are underpinning everything and are quite fundamental. So the University of Oxford is fortunate that we have this amazing resource that is Wytham Woods, which is a 1,000-acre site owned by the university since 1942, just to the west of the city. And actually, we have experiments, long-term experiments running there, which have been running pretty much since then. So the result is it is one of, if not the most studied woodland in the world, from everything from badgers to small mammals to the birds there, to the plant plots, and also insect research, including a couple of new exciting projects, which is what I'm going to be focusing on today. But this will just give you a little flavour of some of the different projects that we have ongoing, from looking at the caterpillars and their phenology and how that impacts on bird nesting, to pollinator surveys, to moth trapping, to work on the grassland areas as well, looking at how droughting, for example, affects these communities. And then two very exciting projects I've been more directly involved with, using modern molecular techniques to research these communities. So before I get onto the projects, I know a very diverse audience that we have. So I just wanted to have a really brief refresh of what we mean when we're talking about some of these different techniques. So the first one, which is quite important, and I know it's been mentioned already by a couple of the talks yesterday, is DNA barcoding. So the concept behind this, it came from the early 2000s, but it's this idea, we know that all these organisms have their genome with all of this genetic information. Is there a region within that which we can use like a supermarket barcode to just identify that species or the species concept to which that organism belongs? And it's a little bit like a Goldilocks system, that we need a region of DNA which varies enough that we can differentiate and discriminate between different species, but it doesn't vary too much, though we can actually find it with our primers when we're looking for it. And we don't also want it to vary too much intraspecifically, so we have these nice, neat species bins for which we can assign. And very fortunately for us, one of the first ones which was proposed, at least for animals, is actually amazing. It works remarkably well. So we use the cytochrome oxidase subunit 1, which is a gene which is in the mitochondria, so it's not nuclear DNA, which is involved in the production of an enzyme which is in respiration, which means it's very, very important. In fact, if there is any mutations here, that would be lethal. So it's remarkably consistent across the majority of the animal kingdom. So for pretty much all animals, that's what we use. There are other markers which we can use, and as well, if we were looking at different kingdoms, we might use different markers or even a combination of different markers. And you see here, I just thought I'd put up an example of one of my favourite DNA barcodes with this lovely Bombus hortorum as well. What a fantastic little bee. And yeah, because these genomes are so enormous, we're talking about millions of base pairs. Actually, the barcode regions that we use are remarkably small. So on average, say one is about 600 base pairs big, which is, if you work it out, back of the envelope, it's 0.0003%. So it's a tiny little fragment, which means it's quite useful. However, DNA barcoding does have limitations. It's only as good as the reference library. And for some taxa, they're remarkably complete. So things like butterflies and moths, actually, we've done pretty much all the species, at least in the UK. But there are other groups and taxa where it's much less complete. So it's very hard to then get your species concept, which you can match to the bin that's produced. Also, for such an important molecule, DNA is pretty crap. And it's constantly just breaking due to the wear and tear of daily life in a cell. But not a problem when we're alive because we have all this DNA repair machinery, which is constantly fixing it remarkably accurately. However, as soon as an organism dies, it starts to break down, and recent studies have shown that actually most DNA degradation happens very quickly. So within minutes of an organism dying, you start to get the large fragments breaking down. And then actually between the mid and the long term, there's actually very little degradation, which does mean because we're using such a small marker region, we can actually sequence the barcodes of highly degraded DNA. So we can sequence things like museum specimens and even subfossils and ancient DNA, which is quite exciting. But also, it is a Goldilocks thing, but it doesn't always work because animals are so diverse just by chance that there's about 5% of animal taxa where CO1 just doesn't work for various reasons, such as maybe the taxa have just diverged too recently, or maybe there just hasn't accrued enough changes there. Or you also have processes such as mitochondrial integration, which can really affect how effective it is. So the first project I'm going to just mention is BioScan, which is part of a global project, but this is the BioScan UK element, where we're doing exactly this. We're barcoding everything. And in fact, BIFoR is one of the projects involved in this, one of the partners involved in this project. Sorry about that. But we're trying to barcode huge numbers of flying insects. So we put these malaise traps out across the UK in all these beautiful, different, diverse habitats, and just as a new method to try and get a handle on the diversity of the species that we have there. And the individuals are then plated up in these plates, and actually we are then bathing them in a lysis buffer and then sequencing the resulting solution. So it's non-destructive, so the specimen remains intact. And this project will not only tell us about the assemblages and the communities of flying insects in these habitats and the way of biological monitoring, but it will also help us to fill in some of the missing gaps in the reference libraries, as well as also be sequencing other associated biological material in those samples, for example, pollen that may be on the insects or even gut contents. So that's barcoding. The next level, and it's quite a big step up, is, okay, we've done this tiny little fragment. What about the rest of it? What's going on for the rest of the genome? So this is where we're doing whole genome sequencing. And the way this works is we start by sequencing lots of little fragments of DNA because that's what we get when we break open a cell. And then we use algorithms to stitch those together and reassemble the correct order. So it's a little bit like doing a jigsaw, but much like a jigsaw with a few large pieces is a hell of a lot easier to do than one with many small pieces. The same is true. So the larger the fragments we can have, so for example, long read sequencing, the much more accurate, especially because DNA is incredibly repetitive. And actually, it's a bit like doing a jigsaw, which is all grey. And you haven't just got one copy, you've got thousands of copies of this jigsaw all put into a big bag, and you're trying to work out which bit goes next to which. So this is what we're doing on the Darwin Tree of Life project. This is the main project that I'm working on, which is an incredibly big and ambitious project. We want to sequence the full genome of every single species of eukaryotic organism in Britain and Ireland, which, based on UKSI, is more than 70,000 species. Not only are we sequencing them, we're also producing very high quality sequencing because this is all based on long read sequencing. Such a big project involves many different partners, from experts in biodiversity to sequencing to bioinformatics. But why are we doing this? Well, the first answer is, well, because we can. This is the first time in all of human history where actually a project like this is feasible. But just because we can do something doesn't mean that we should. So what are the other reasons for the motivations for producing all these reference genomes? Well, I categorize those in two different camps. The first is discovery science, and actually we don't know what we don't know. By looking at these genomes, we can discover all sorts of relationships and biology that we had no way of elucidating before. And then the second is enabling science, whereby genomes, high-quality reference genomes, are quickly becoming an essential component of the 21st century biology toolkit, which is a really grandiose sentence, which I love to say. It's very exciting. For what are some of the examples for how we can use this toolkit? Well, first of all, we can resolve phylogeny. So there are different taxa where it just doesn't work by using barcoding or even using multiple markers. Well, we can do whole genome alignments, and we can actually get to the bottom of some of these tricky phylogenies. We can also look at the actual structure of the genome itself, such as things like karyotyping. We can then, genomes are by far the best way to study gene and gene family evolution as well, which is something that we've been doing quite a bit off Oxford. A reference genome is also an essential component to be able to do population genetics. You need that initial reference genome to then highlight your marker region. So, for example, large blue butterfly, which we got a permit after much work to go and collect and sequence. The genome that we've just produced for that is going to be so useful and helpful for us trying to do population genetics to try and map the genetic variability as the reintroduced populations spread. And then finally, it's also quite exciting prospects for biodiscovery. There's all sorts of physiology going on in organisms which we don't know about that the genomes can help us to elucidate. So my work specifically is in sample acquisition. So it's all about what species do we have in the UK? We can't sequence everything at once. So what are our priorities? What are the species which are particularly interesting, iconic, or economically important? Then we go out and we collect these species, and then we have to identify them and prepare them. Because we're doing long read sequencing, we need to flash freeze from live, which is certainly a challenge. And then once the genomes are produced, they're all released publicly. In fact, our project is completely open. So for every stage, all of our data is open and available for anyone to come and use, with the caveat is this is a draft. And then when we produce a genome, we create a genome note, which is a data note publication announcing that, for which we also write a little biological background just to try and a bit of natural history and the biology of the organisms. We're at the stage now where we have so many genomes coming out, we can't possibly write all of these genome note introductions. So if anyone's interested, please write some genome note introductions for your favorite organism that we've sequenced. We would love to have contributions.
Carry on.
Yeah, the Oxford specific part of it is all based primarily at Wytham Woods, which is a genomic observatory where we want to sequence every single species within the woods and put it in the context of this historical and well-studied site. And this is focusing primarily on arthropods and other invertebrates, although we have sequenced some things like badger. And done quite well so far. So over the past 5 years or so, I've collected over 5,000 specimens of about 2,500 species. And you see some pretty ones here, because why not? And actually, the rate of species discovery is continuing. Is continuing to find new species all the time. You think actually such an intense look at a site, we might quickly run out of new species to find, but that's not been the case. So we see here the kind of cumulative number of novel species and families discovered per sampling day, which is quite a reassuring sign. And when you're looking at a site so intensively, you inevitably discover exciting things. So here's some of the new species that I found for the site, or some interesting rediscoveries of things which have not been seen for a very long time, like the emperor moth, which hadn't been seen for 30 years at the site. I actually reared that from a caterpillar, which was quite fun. And then we find the really interesting stuff, the real scarcities that get people like me really excited. So this was this lovely Pandavirula fly that decided to land on a log in front of me and nearly make me fall over. Because it previously was only known from 2 localities, Windsor and South Gloucestershire and Worcestershire. So what's it doing in Wytham? It's nowhere near any of those sites. Was it always there and we just missed it? Is it moving and spreading? Who knows? And then we get really, really good stuff. Or actually school kids that come and help find really, really good stuff. So I was doing a public engagement workshop with a local Oxford school, and one of the boys came up to me and said, "Oh, I found a beetle." I was like, "Great!" I'm expecting kind of one of these really common ones that you see, and he shows me this beetle, and I was like, I honestly don't even know what that is. And that's where I get quite excited, because I know most of the UK longhorns. I know all the UK longhorns, but I'd never seen this before. And then it turns out it was, well, initially we thought it was the 4th English record, but then I had a look on iNaturalist and another one turned up. So yeah, the 5th English record of this European species, which is very exciting. And we're now publishing a small note on this, for which the school are going to be co-authors, which is a nice thing to do. So that just leaves me to acknowledge there's far, far, far too many people to list. This is an acknowledgement to all the people involved with these various projects. Thank you.